T-cell engagers are off-the-shelf therapies that may overcome some of the limitations associated with CAR T-cell therapy. Several CD20, CD3 T-cell engagers are currently being developed to treat B-cell lymphomas, and the T-cell engagers show the clinically meaningful efficacy. However, therapeutic options remain limited for patients who relapse after or are refractory to CD20, CD3 T-cell engagers, and the loss of CD20 expression represents a significant clinical challenge to CD20, CD3 T-cell engagers use. In addition, treatment options are limited for patients who relapse or are refractory after CAR T-cell therapy, as well as for those who are ineligible for CAR T-cell therapy. Surovatamig, formerly known as AZD0486, is a novel IgG4 fully human CD90 CD3 bispecific T-cell engager with attenuated CD3 binding to minimize cytokine release. We presented safety and efficacy data from the phase 1 first-in-human ongoing dose escalation of surovatamig in patients with refractory relapsed DLBCL. And enrolled patients received fixed duration surovatamig intravenously for 2 years. As for safety data, in 77 patients who received surovatamig in double step-up dosing, any grade CRS and ICANS were observed in 49% and 20% of patients, respectively. As for CRS, all events were grade 1 or 2, and the median time to first CRS and the median time to resolution was 1 day each, and 27% of patients received tocilizumab. Grade 1 to ICANS event were observed in 10 patients, and grade 3 ICANS occurred in 4 patients that were transient and reversible. Median time to first ICANS and median time to resolution was 1 day each, and no seizures were observed. Overall, most of all adverse events were manageable, and safety profile of surovatamig were acceptable. As for baseline patient background, more than 70% of patients had a history of refractory to last line of therapy, and more than 40% of patients received prior CAR T therapy, CD19 CAR T therapy, and 13% of patients received prior CD20 T-cell engagers. So that is to say, most of enrolled patients had high-risk features and heavily pretreated disease. However, surovatamig at target doses 7.2 milligram or higher showed promising efficacy, such as overall response rate 74% and CR rate 63% at the target dose of 25 milligram, including the CD90 CAR T exposed patients. The median duration of response and the median duration of CR were not reached, and 12 months estimated DOR was 77%, and the duration of CR was 87%. Even though the limited follow-up duration with a small number of patients at this point, there seemed to be a potential plateau in duration of response and duration of CR. So, surovatamig has demonstrated high efficacy and a manageable safety profile in relapsed refractory DLBCL patients. As a novel CD90 targeting T-cell engager, surovatamig is expected to play an important role of DLBCL treatment in near future.